Hi everyone, and welcome to our video on regulation of gene expression. This is part of our Module 6 Part 1 series about cellular control, and what we'll be looking at in this video is specification reference 6.1.1b, the regulatory mechanisms that control gene expression at the transcriptional level, post-transcriptional level, and post-translational level. First thing then, if we think back to some basics on cells, then in our cell we have a nucleus, and the nucleus contains all of the genes that exist within that particular organism. Now, one thing that we do have to remember though is even though all of these genes are present, they are not all expressed. So what we actually have is a situation where we need to make sure that certain cells express the right genes at the right time. So basically that means we need some kind of a regulatory mechanism that is either going to switch genes on or off, depending on if they're needed or not. In terms of what we have as regulatory mechanisms, there are three types: transcriptional regulation, post-transcriptional regulation, and post-translational regulation. And hopefully we are familiar with at least the two of those words there, transcription and translation. If we think back to the work you did in Module 2 where you were having a look at protein synthesis, transcription is that formation of the mRNA, messenger RNA strand, and translation is where that messenger RNA is being read by the ribosomes in those triplet codons. And therefore we're able to synthesize the polypeptide chain using transfer RNA to bring in the correct amino acids. So if we start off with thinking about prokaryotic cells and the process of transcription, if we think about the prokaryotic genome, then what we have are a range of genes, some of which are going to be coding for enzymes for their basic metabolic reactions, for those basic cellular functions, and others will be coding for enzymes that are required for certain conditions. So what we end up with is a bit of a split. Any of these enzymes that we need for the basic cellular functions will be synthesized at a pretty consistent rate, just because those reactions are going to be happening at, again, a pretty constant rate. So clearly, if we know that the reaction is happening at a specific rate, then the enzyme will be required at a set value. If, however, we're talking about an enzyme for a reaction that only happens at certain times, then we don't need this continual synthesis of the enzyme. We need to basically be able to vary how much of that enzyme is present based on those conditions. So when we need more, we create more. When we need less, we produce less. So what we're going to have a look at as far as our transcription control in our prokaryotes is something called the lac operon. Before we delve into the finer details of this, let's make sure we understand two terms that are going to come up here. First one, structural genes. These are the genes that code for a protein that's got a cellular function. Things like an enzyme, for example. So your enzymes are going to have a particular function within the cell, and therefore they are a structural gene that codes for them. The regulatory genes are going to be coding for proteins that control the expression of those structural genes. So basically, if you think about what regulation is, it's all about controlling things. So regulatory genes actually code for a protein that controls the expression of the structural genes. Now, the lac operon is this regulatory control at the transcriptional level. And we're using this phrase operon, and whenever we talk about that, it's just a group of genes that are functioning as a single transcription unit. So basically, these genes are all controlled by the same promoter. We're going to be looking at E. coli then. So E. coli is a bacteria commonly associated with food poisoning, but we're going to be focusing on this operation, the lac operon, within E. coli. So the normal situation for E. coli is that it's going to be utilising glucose as its respiratory substrate. But there will be situations where glucose isn't available. Now, in that scenario, if glucose isn't available, but another sugar, lactose, is present, then what E. coli actually does is it hydrolyzes the lactose because obviously lactose is a disaccharide, remember, and the products from that hydrolysis reaction are then going to be used as respiratory substrates. So the way we can do this is by producing certain enzymes, but these enzymes are only going to be made when lactose is present. So the lac operon then is a 6,000 base pair long length of DNA within that bacterial genome. And basically it looks like this. So what we've got here then is the little section of the DNA that is the lac operon. So the P regions are promoter regions. This I is our regulatory gene. We then have three of these things that have lac in front. So we've got lac-O, which is the operator, lac-Z and lac-Y, which are both structural genes. So lac-Z is going to be coding for beta-galactosidase, which is obviously an enzyme, and then lac-Y is going to be coding for lactose permease. So what we've got, two structural genes, both coding for an enzyme that has a particular function. Lac-O is the operator region, and then the I, the regulatory gene. So, in terms of what these enzymes do, lactose permease is going to allow lactose to enter the bacterial cell wall. If you actually just break it down, it kind of tells you that in its name. So lactose obviously to do with the lactose that is going to allow entry, and permease sounds like the word permeable. And if something is permeable, it obviously allows it to cross a barrier. So obviously the ending ASE tells us an enzyme, so lactose permease just makes the bacterial cell wall able to let lactose cross it. Then we've got our beta-galactosidase, and what we have is an enzyme that is going to be hydrolyzing lactose into the two monomers it's made from, glucose and galactose. So let's have a look and see how this actually works. And we've got two situations we will look at. The first is when no lactose is present. So in this situation, when there's no lactose, we will be transcribing the regulatory gene, this I. Now, the regulatory gene is going to then produce the lac-I repressor protein. So basically, that little regulatory gene produces a repressor protein called lac-I. The lac-I repressor protein is then going to bind to our lac-O operator region. Now, if you imagine that we've just bound something, I'm just going to draw a blob for the sake of it. So we've just bound something onto lac-O there. When we actually want our RNA polymerase to come in, it's not actually going to be able to bind to the promoter region because that lac-I repressor protein is kind of blocking its access. So because we can't get the RNA polymerase binding to the promoter region, it will not then transcribe our lac-Z or lac-Y. If we now consider the situation when lactose is present, the lactose is going to bind to this lac-I repressor protein that's been made. So we've got this repressor protein that's then going to bind to lactose, and as a result of that, it alters the three-dimensional shape of that repressor protein. That means it can't bind to our lac-O. So we're not now blocking the promoter region, which means that our little RNA polymerase can come in, bind to the promoter region, and then it's going to be able to move along and transcribe lac-Z and lac-Y, which means we're then going to be able to produce those enzymes. The next place that we need to consider our transcription regulation is in eukaryotic cells. Now, when we think about our cells, they all contain these things that are called housekeeping genes. And these are the genes that are needed for all these basic cellular functions. So all cells are going to express these housekeeping genes because they are literally giving us the basics for cells to function. What we then have are things which are called transcription factors. And a transcription factor is a protein or a short non-coding piece of RNA that's going to control which genes are switched on and which genes are switched off. And these make up around 10% of your genes. The way in which they work then is they're going to bind to a specific region of the DNA, those promoter regions, and then when they've bound, it will either be aiding or inhibiting the binding of the RNA polymerase to the DNA. So obviously if it's inhibiting, that means that the RNA polymerase is going to be prevented from binding, and therefore that gene will be switched off. If, however, it aids in the binding of the RNA polymerase, then we will be expressing that gene, so it's switched on. So purely by having these transcription factors that can bind to those promoter regions, we will either switch the gene on or switch them off. And this is obviously critical when it comes to the ability to specialize our cells, because to actually have those differentiated cells that carry out a particular function, we need to ensure only certain genes are going to be expressed, so they're switched on. The other ones need to be switched off. And this is how we do it with the use of those transcription factors. One thing that you would have mentioned back in module 2 was having a look at that cell cycle regulation. So determining when we're actually going to create more cells through that process of mitosis. Now, what we do find is that we've got a particular type of gene called tumor suppressor genes that are all involved in regulating cell division. And the important thing here is that if we've got one of these tumor suppressor genes that has a mutation, this is then going to obviously prevent that regulation of cell division, and therefore it will lead to a cancer. So what we can actually see here is tumor suppressor genes, they're basically stopping tumours, because a tumour at the end of the day is an uncontrolled cell division that's occurred within the body. Therefore, these genes are suppressing the formation of a tumour. If a mutation occurs in them, tumours may occur. Okay, let's move on to our second type then, the post-transcriptional gene regulation. If we think about the structure of a gene, then basically we've got different regions. There are introns and there are exons. So the introns, which are the red bits on our little diagram, are the non-coding regions of DNA, and the exons, which are the blue little sections, are the coding regions of DNA. So when we actually look at our actual gene here, you can see that the exons are basically being split up by these introns. So coding regions split by the non-coding regions. When it comes to forming our primary mRNA, then all of the DNA is going to be transcribed. So that's the introns and the exons. Literally, we're just creating that mRNA strand of all of it. And that's what we term the primary mRNA. But that's not actually the end of our mRNA's production. What we need to do is edit that mRNA strand to make it so that all we have are the exons joined together. So the way this happens is the primary mRNA is going to have the introns removed, and then the exons just get spliced together. And quite often this is going to involve a type of enzyme called endonuclease enzymes. It's not a case of we've cut these sections of the mRNA out and that was a complete waste of generating them, because these introns that are being removed can be involved in gene regulation. So what that means is that once we've removed those introns, they may actually have a role in regulating certain other genes within the cell there. And the way in which the exons are then going to be spliced, so joined together, will alter the protein that we code for. So what that actually means is that that one gene that we've transcribed from the actual DNA template into the mRNA, that primary mRNA can make more than one mRNA that allows us to code for proteins. Because if you imagine that we've got, if I just number these little pieces down here, 1, 2, 3, 4 little sections, so those are our 4 exons, the coding regions, we could obviously join them 1, 2, 3, 4. We could also join them as 2, 1, 3, 4. we could join them as 3, 4, 1, 2, and so on. I'm not going to do them all, it will take a while. But because we've actually got the ability to create these small sections of the coding DNA and then join them in that splicing process in different ways, that single gene actually has the ability to code for more than a single protein in the organism. The last category of our gene regulation is post-translation. So post-translation is basically about activating proteins. So this typically involves a process of phosphorylation. And the example we're going to look at to explain this is cyclic AMP or CAMP. This is something that will come up in module 5 as well. So it's a nice little repeating little molecule. Okay, so the first thing that happens is we're going to have some kind of signaling molecule binding to the receptor on the plasma membrane of our target cell. This could very well be a hormone, for example. As a result of that binding to the receptor, we will activate this transmembrane protein, which activates a G protein. So what we've got, transmembrane protein, is obviously a protein that spans the membrane there, and that is then going to activate another type of protein, these G proteins, which act kind of like a switch in our cells. That G protein activates another enzyme, adenyl cyclase, and the adenyl cyclase catalyzes the conversion of ATP into the cyclic AMP. Now, once we've got our cyclic AMP or CAMP formed, then the next thing that's going to happen is it activates this stuff called pKa. Now, you don't need to know what it stands for, just pKa is fine. If you want to, it's a stuff called protein kinase A. Now, pKa then catalyzes the phosphorylation of various proteins, which means we're then able to activate other enzymes. So what we can see here is that phosphorylation process means that we're able to activate other enzymes in the cytoplasm and therefore carry out other reactions. Another example of what the pKa is going to do is it phosphorylates this stuff called CREB, so cyclic AMP response element binding. CREB is fine. And the CREB enters the nucleus, which is where it then acts as a transcription factor. So that means it's going to regulate the process of transcription. So what we have actually looked at are those 3 different categories of how we're able to regulate gene expression in both prokaryotes and in eukaryotes. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded, and head on over to the website where you can find a range of other resources to help you in your A-level biology studies.